Welcome to our lesson on gene expression, and we'll take a look at RNA. The information that is inside of DNA is all the information needed to build a whole entire organism, even a very complicated organism like a human being. So take a look at this picture on the right. It all begins with things known as chromosomes, and we know that we humans have 46 chromosomes. We know that prokaryotes have only one chromosome. We know dogs have like 78 chromosomes, and chromosomes are made up of DNA. And DNA is very, very, very long, and there are pieces of DNA that we call genes. And genes are pieces of DNA that actually code for a specific protein. All right, but it's not so simple. So what actually happens is inside the chromosomes is the DNA, and the DNA are these genes, and the genes are then going to be copied. You're going to make a copy of them in something known as RNA, and that part is new. We haven't talked about RNA before. So we take that RNA, which has the message that's inside the DNA, and take that message to the ribosome and use it to build a protein. Why do we want the protein? Well, it's proteins that are going to allow you to express a specific type of trait, like, for example, eye color. You can have green, right, or blue, or brown, or hazel, or a combination or blend of those different types of eyes. But the reason you have these eye colors is because of proteins. And if you remember proteins are a very diverse group of biological molecules that cause all these different things. Not only do they cause physical traits like curly hair and straight hair and dark skin and light skin, they also make things like insulin, for example, that helps you regulate your blood sugar. Or, for example, actin and myosin, which are inside of your muscles that actually allow your muscles to contract and work and move your skeleton along so you can go to the places you want to go. So proteins really run the show in our bodies. But how do we make proteins? That information is inside of our DNA, and that genetic information in the DNA we copy it into a form known as RNA, and that RNA is used inside of our ribosomes to build proteins, and proteins cause these traits to be expressed. So a little bit of flashback to our protein lesson. Proteins are made of very, very long chains of amino acids. Amino acids are the protein monomers, and you have 20 different amino acids that arrange themselves in all different orders to make a huge variety of different types of proteins. And I want you to familiarize yourself a little bit with these amino acids, again, because they're making a big comeback in this chapter. So look at some of the names: lysine, histidine, tryptophan, right? So these are going to arrange themselves in all different kinds of orders to make specific types of proteins. So amino acids have some main components, and this isn't new, this is a review. There's an amino group in each amino acid. There's a carboxyl group in each amino acid. There's an H group. And then there's something known as a side chain. And that side chain is variable. It varies from amino acid to amino acid, and it gives the amino acid its unique traits. So if you go back to this chart of our 20 amino acids, you can see that most of the amino acid is the same, right, going from one amino acid to the other, but this highlighted area is the side chain. That's different between each amino acid. Okay, so a chain of amino acids is actually called a polypeptide. So here are your little amino acids, and they link up with peptide bonds to form a polypeptide. And once that polypeptide twists and turns and gets into a complicated three-dimensional shape, then we call it a protein. All right, so proteins are formed when these polypeptide chains twist and turn and combine with each other and make this complex three-dimensional shape. All right, so this is how things really happen, and we're going to introduce some scary terms that will get easier as time passes and you get more familiar with them. So it all begins with DNA, and a part of DNA, a gene, is then it undergoes a process known as transcription. And in transcription, we take the DNA, we take that gene region, and we use the DNA as a template, as something we're going to use as a model to create a strand of RNA. Okay, you want to think of the DNA as something that's like really precious. It's like our master copy, right? And you want to keep it in your nucleus. It doesn't go anywhere. So if we want to take information from the DNA, we've got to make a copy. The copy is movable, and that copy is called the RNA. And the RNA can go to the cytoplasm and find a ribosome, which incidentally, if you remember, is a protein factory. And that ribosome can then decode that message, that secret message that's inside of RNA, and use that message through a process known as translation to build a protein. And you can see this protein here is made up of these little circles. Those are each amino acids linking together to form the protein that we wanted. And this could be any type of protein. Maybe this protein is some insulin, right? Because you need to regulate your blood sugar. Or maybe it's melanin because you spent a lot of time in the sun. It's time for you to tan. Okay. So what is RNA? Because we haven't really discussed it yet. RNA is a nucleic acid like DNA is. So it's made up of nucleotides, but it's single-stranded, unlike DNA was. RNA nucleotides have a couple groups. They have a phosphate group. They have a sugar, but this time the sugar is not deoxyribose. This time the sugar is called ribose, and RNA is ribonucleic acid. It's named after that sugar. And the nitrogenous bases in RNA are almost the same as DNA. We have adenine, which we symbolize with A. We have guanine, which we symbolize with G. Cytosine, which we symbolize with C. But the last one is not thymine. In RNA, we have uracil instead. Now, C still bonds with G, but A bonds with U, with uracil. So let's compare DNA and RNA side by side. So you want to think of, first of all, DNA as like your master copy. It's really precious. It stays in the nucleus. And you want to think of your RNA as just like a copy, like a blueprint of the master plan that's movable and usable. And it's not as precious and it's not as long. It's just a piece of the DNA that you need at that given time. Now, the sugar that's involved in DNA is called deoxyribose. The sugar in RNA is called ribose. We'll look at a picture of DNA and RNA side by side next, but DNA is double-stranded. It's the double helix. RNA is single-stranded. DNA has A, C, G, and T, whereas RNA has A, C, G, and U. So if we look at DNA and RNA side by side, you can see the DNA is a twisted ladder and it's got 2 strands, and RNA has only 1 strand. And then you can see the different nitrogenous bases here, and you can see that here we have uracil, whereas in DNA we have thymine. So that's one important change. Okay, so there are 3 types of RNA. There's messenger RNA, which we call mRNA for short. And messenger RNA copies the DNA's code and actually carries that information to the ribosomes. Then you have something known as ribosomal RNA, or rRNA for short. And this actually helps to build the ribosome. It makes up the ribosome. The ribosome is made up of ribosomal RNA and protein. And the 3rd type of RNA is called transfer RNA. This is called tRNA. And this is going to bring in the amino acids so that we can build the protein, so we can build the polypeptide. And we'll see each of these one at a time. So the first one is messenger RNA, of course. And this is actually a long, straight chain of nucleotides that's made in the nucleus. And what it does is it copies the DNA and it leaves the nucleus through these nuclear pores. It contains the bases, of course, of A, G, C, and U. There's no T's. And it carries the information for a specific protein. It's made up of 500 to 1,000 nucleotides. So a little bit more about messenger RNA. There are these things known as codons. So mRNA is made up of a sequence of 3 bases called codons. So, and there are many codons inside of mRNA, and every 3 letters inside of mRNA stand for an amino acid or a stop signal. So each codon stands for a type of amino acid, or it tells basically the ribosome that to stop copying because we're finished making a protein. So you can see here the mRNA's got all these letters on it, right? This is its sequence that it got from the DNA, the secret message that it got from the DNA, if you will. And every 3 letters is a codon. So this is a codon, the next 3 is a codon, the next 3 is another codon, and each codon stands for something. So the first one is methionine, which is actually the start codon. It always starts the sequence. The second one, this blue one, is valine, this is serine, this is proline, and then all of a sudden we hit a stop codon, which means we're done. And we can actually hook up these amino acids to each other to form a chain, and then this can be a complete polypeptide. So that's how mRNA works. Now, we can actually discover or figure out what each one actually means. So this is actually like a little code reader. It helps us break the code that's inside of RNA. figure out what it means. And you use this by starting in the middle and working your way out. So for example, we want to figure out what AUG means. So we have to start in the middle. So in the middle, I find A. A is right here. So I'm ready to go, and I have to now stay in this quadrant. I have to stay here. And then the next letter is U. So I'm going to go out one level, and I'm going to look for U. And U is right here. And the third letter is G, so I can go out one more and look for the G, and the G is over here. So my amino acid that AUG stands for is methionine or methionine. Now we're going to try a couple other ones together. So what about GGG? So again, I'm going to start in the middle, G, work my way out, find the second G, and then I'm going to go out one more, and I'm going to look for the G, and that's right here. That's glycine. Okay, so let's try another one. CAU. What could CAU be? So we start in the middle, C, go out one, A, and we look for the U right here, histidine. Okay, one more, UGA. UGA, and it says stop. So stop is not an amino acid. It just means basically stop translation, which is the process that we're doing when we're making sense of the information in RNA and using it to build a protein. Okay. So ribosomal RNA is basically what, along with proteins, makes up the ribosome. So here is ribosomal RNA, and here's a nice close-up of what a ribosome is like. If you remember ribosomes, they're protein factories, and these are ribosomes right here. Some are free ribosomes, and a lot of them are actually attached to the rough ER. That's why the rough ER is rough, because it has all these ribosomes attached on it. Okay, so our RNA is a single strand. It's 100 to 3,000 nucleotides long, and it's made inside the nucleus of the cell. And the ribosome is where protein synthesis actually happens, so where we build the proteins. Okay. Now, transfer RNA is the last one. It has a clover leaf shape, like if you've seen a clover plant before, okay? And it's a single-stranded molecule with attachment site at one end for an amino acid, right? So it's got an attachment site for an amino acid right here, okay? And the opposite end has 3 nucleotide bases called an anticodon or anticodon, which is right here, okay? So what is an anticodon? Because we learned about the codon. The codon's on the mRNA, right? GUCCAG, right? That's the codon. And it turns out that's on the mRNA, but on the tRNA are these things known as anticodons. So an anticodon is 3 bases that are complementary to the 3 on the codon. So for example, if the codon is ACU, the anticodon would be UGA. So here's another codon, we have GAG, so we're going to use base pairing rules to figure out what the anticodon is. So with G, we would have the C going with that. With A, we would have not T, but U going with that, because now we're working with RNA. And with G, we would have the C going. So you can see that the codon and the anticodon are complementary. And the tRNA is actually going to have an amino acid attached to it that it's going to bring in so that we can start to build a protein. So I know that at this point it's probably pretty confusing, and it'll be a long time before all of this makes sense. So here's the ribosome again, just so you could see that. This is the mRNA strand, and this is the tRNA, and see how the tRNA is carrying the amino acids in, and it's building a chain. In any case, I know this sounds pretty complicated at this point, but we're going to work through it, and it's going to make more and more sense. Today it was more of an introduction to how genes work to make proteins, to express traits, and how RNA works, and the different types of RNA that there are. We're going to take a close look at transcription and translation in the coming lessons. Thank you so much for listening.